Hi, my name is Brian Kim, professor and vice chair of research in dermatology, director of the Mark Lovell Center for Neuroinflammation and Sensation at Icahn School of Medicine at Mount Sinai.
Vignette 8, AD, PN, and CSU, shared inflammatory pathways, distinct itch. Previously, we explored the distinct cellular and molecular repertoire of type 2 inflammation, allowing us to distinguish dysregulated type 2 immune responses from dysregulated responses in type 1, for example, alopecia areata or vitiligo, or type 3, for example, psoriasis. We also discussed how type 2 cells interact in the skin and highlighted how immune dysregulation can imprint unique responses even within the same organ system, leading to the characteristic signs and symptoms of different type 2 inflammatory diseases. In this series, we focused on various cellular and molecular interactions with sensory neurons that generate itch and trigger neuroinflammation in 3 chronic pruritic inflammatory skin diseases: AD, PN, and CSU. While type 2 inflammation underlies all 3 conditions, understanding the nature of the crosstalk and roles of the cells and mediators involved helps define the characteristics of their itch and prevailing symptoms. For instance, type 2 cells and the cytokines they release, such as IL-4, IL-13, and IL-31, play fundamental roles in itch underlying each condition, but do so by targeting distinct cellular and molecular components therein. For example, while IL-31 acts as a direct pruritogen, IL-4 and IL-13 can directly stimulate sensory neurons and cause neuronal sensitization, increasing the sensitivity of itch processing neurons to pruritogens like IL-31 and histamine, promoting further itch and scratching behavior. However, factors like the spatial temporal sequence that cells and mediators follow to act on pruriceptors and trigger itch likely differentiates these conditions' clinical presentations. Itch in AD is attributed to multiple cellular and molecular mechanisms, with a major initial contribution from epidermal barrier dysfunction. Subsequent scratching results in excoriation and epidermal barrier thickening, perpetuating inflammation and itch sensitization. The nerves that transmit itch are primarily, though not exclusively, sensitive to non-histaminergic or histamine-independent itch pathway activation, which underlies chronic itch. However, most patients with AD express high levels of antigen-specific IgE, which may be associated with itch flares in these patients. The cause of PN is not yet fully understood, but neuroimmune dysregulation appears to be a primary driver of its pathogenesis. Sensory neuron activation, whether through abnormal innervation or neural hypersensitivity to itch stimuli, can create an initial itch sensation, driving maladaptive scratching behavior, which may result in aggravated immune dysregulation through mechanical skin trauma and the release of pruritogens. Neuronal activation also leads to neurogenic inflammation, creating a pro-inflammatory tissue microenvironment. Type 2 cytokines IL-4, IL-13, and IL-31 directly stimulate and enhance neural activation at sites of inflammation, amplifying itch sensation. These cytokines also contribute to dermal fibrosis through extracellular matrix deposition, fibroblast proliferation, and myofibroblast differentiation. PN's prolonged itch-scratch cycle promotes epidermal stress responses and robust dermal tissue remodeling and fibrosis, thought to manifest as its characteristic nodules. Thus, while AD may start with barrier dysfunction and inflammation, PN may start with itching. CSU stems from dermal signaling and is closely linked to mast cells and basophils that generate both histaminergic and non-histaminergic itch through the release of inflammatory mediators like IL-4 and IL-13. Both the classical histaminergic and the more recently recognized non-histaminergic pruritus arising in the dermis help distinguish the itch, burning, and pain in CSU from AD and PN. CSU's wheals, angioedema, itch, and burning develop following mast cell and basophil degranulation via IgE-dependent and IgE-independent pathways. While AD and PN involve cytokine-driven itch, CSU likely involves a variety of additional intermediates, including histamine and other factors. Although type 2 inflammation and itch link AD, PN, and CSU, they are distinct diseases. Emerging data suggests that the types of signaling involved dictates the characteristics of itch, while signaling dysregulation determines the disease-specific nature of itch. Differences in itch quality across the conditions likely influence how patients scratch, for example, intensity, frequency, and duration of scratching, rubbing, picking, poking, or applying pressure to alleviate the itch, as distinct pruritogens induce different scratch durations and patterns. Moreover, the particular form of scratching may impact the typical lesions associated with these conditions, such as excoriated plaques in AD, hypertrophic nodules in PN, or absence of lesions in CSU, and may be suggestive of the type of itch that is evoked. AD and PN's continuously chronic itch creates the persistent itch-scratch cycle that damages the skin and contributes to each condition's characteristic lesions. In contrast, the itch in CSU may be more discontinuously chronic, with patients experiencing relatively itch-free moments despite having itchy wheals that recur intermittently for 6 weeks or longer. In this respect, the chronicity in CSU may be more reflective of an ongoing series of histamine or other factor-mediated acute itch episodes. In summary, AD, PN, and CSU are distinct diseases, each exhibiting unique characteristics from variable itch quality to underlying pathophysiology and skin manifestations. Although each is rooted in type 2 inflammation, the difference in similarities across all three are a testament to the fine balance and crosstalk between the cells, molecules, and neurons responsible for their expression.